Punnett squares are a tool that's used to predict what's going to happen in a Mendelian cross, like we're going to be doing a lot in this chapter. And so I'm going to walk you through how to do one, and then we'll walk through another example, and then we'll talk about a test cross. So we'll get some practice using these Punnett squares. So the way that genetics problems are going to typically work is there's going to be a word problem. So it's going to describe the parents, and you'll need to sort out from that what the genotypes of the parents are. So maybe the question would say, two heterozygous tall plants are crossed. Predict the offspring. So again, there's going to be some sort of word problem where you need to apply what you know to come up with the genotypes of the parents. And this is a pretty easy one. Since both parents are heterozygous tall, then both parents must be big T, little t. So one parent is big T, little t, the other parent is big T, little t. The next step is to decide which gametes those parents can make. So again, this is pretty straightforward. In this case, the male can either make a gamete with a big T or with a little t. And so same with the mother, because she's the same. Her egg either has a big T or a little t in it. So then you create a box. And so for a single factor or a monohybrid cross that's only involving one trait, you just make a box with 4 squares. And then you write one parent across the top, their gametes. So I'll do the sperm on the top. So I'll put a little tail so we know it's the sperm. And then the egg on the side. And then in the middle of the box is what happens if that sperm and that egg combine. So this sperm and this egg combining would give us an offspring with big T, big T. If this sperm combines with this egg, we'd get this. If this sperm got with this egg, it would be this. And if this sperm got with that egg, it would be this. So now we interpret our Punnett square, and we see that there is 3 tall offspring to 1 dwarf offspring. And that's what's called the phenotypic ratio, because it's about phenotypes. You can also do a genotypic ratio. And in this case, it would be 1 big T to 2 big T to 1 little t. So it puts out all the different genotypes in a genotypic ratio. So let's do another one together. Purple is dominant to white color in pea plants. A white pea plant is crossed with a heterozygous pea plant. Use a Punnett square. So the hardest part of these problems is usually getting from this word problem to the correct genotypes of the parents. So we have to remember dominant and recessive relationships and what the terms homozygous and heterozygous mean. So heterozygous is straightforward. That means there's 2 different. So that means one parent has a big P and a little p. The other parent is white. Because white is recessive, the only possibility is that the white parent is homozygous recessive. So those are the genotypes of my parents. This one can make a sperm with a big P or a sperm with a little p. And if this is the female, she can only make eggs that have little p. So now I set up my Punnett square. And again, put my sperm up here. And my eggs here. Combine them here for what my offspring would be. And I see that I would get 2 to 2 of purple to white. And this can also, it's just like fractions, it can be reduced 1 to 1. So 2 to 2 is the same thing as 1 to 1. And then I have 0 big P to 2 big P to 2 little p. So that's my genotypic ratio. And again, this can be reduced, but just for simplicity, I've drawn it out as the actual numbers from the Punnett square. So another kind of cross is called a test cross. And this has to do with something we were just talking about. When you know that an organism is recessive, you know that it must be homozygous. So if a flower is dwarf, it must be little t. But if it's tall, and that's all you know about it, it could be big T or big T little t. And so if you just walked up to some flower one day and you didn't know what its parentage was, you wouldn't be able to tell if it was homozygous dominant or heterozygous. So one way to figure that out is a test cross. A test cross lets you figure out the genotype of an organism when all you know is the phenotype. So basically, consider that if you had the big T individual, right? So this individual, then they can only make the big T gametes, right? If you cross them with the little t individual, so look down here for the Punnett square, you can see that I would get all tall offspring. But if I took that, the same unknown, so this is my unknown tall plant, if it was big T little t, then this is what the Punnett square would look like. So taking big T and crossing with little t. So this part's the same. Then I would get 1-to-1 tall to dwarf. So I can perform a test cross to find out, does my unknown give me all tall offspring, or does my unknown give me 50% dwarf offspring? If I get 50% dwarf offspring, then the unknown individual must be a heterozygote. If I get 100% tall offspring, then my unknown individual must be homozygous dominant. So take a look at this question. Purple is dominant to white color in pea plants. A purple pea plant of unknown genotype is crossed with a white pea plant. 400 offspring are analyzed. 189 are purple and 211 are white. What must be the genotype of the unknown purple pea plant? So basically, they're performing a test cross. The purple pea plant, if it's purple, can either be homozygous dominant or heterozygous. If I perform a test cross, that means crossing it with the homozygous recessive individual. If it's big P, carry out that Punnett square, and I cross with the homozygous recessive, then I will get all purple offspring. If it's a heterozygote, so again making a little mini Punnett square here, and I do a test cross, then I will get 1-to-1 purple to white. So if I look at the data, they counted 400 offspring, 189 are purple, 211 are white. So this isn't exactly 1-to-1, but it is approximately 50% white. So this best matches this data. So my individual, my unknown individual, must be a heterozygote. And that's how test crosses work.